Hello everyone. Today I would like to have a brief introduction about my research topic, estimating transpiration and evaluating the controlling factors of Larix species are necessary for the water management in the forested area in Northern Hemisphere. Our research have two main objectives. First, estimating the large transpiration, and the second one, evaluating the factors that control the variability of transpiration. To develop the calibrated equation, an experiment was conducted in the laboratory using one sample tree. In the field, 6 trees were installed sensors to measure safflower density, while the climate factors were monitored using a climate station. Based on experiment results, we found a new equation, which is different from the original equation proposed by Ranier. The application of new equation may reduce the underestimation by 28%. As the first step of analysis, we examine the variability of safflower density using 3 days with different environmental conditions. 3 safflower densities varied among simple trees. In our study, the variation in safflower density did not depend on tree diameter or sapwood area, but it may relate to canopy position, the leaf area index of the disease such as the larch canker. We also found that safflower density of all sample trees followed similar trend from daytime to nighttime. Although the external conditions varied a lot, there was no big differences in safflower density among selected days. During the day, all trees had the same responses to environmental factors, which shaped the way how safflower density changed from daytime to nighttime. The relationship between transpiration and environmental factors was clearly shown in fully development period. Transpiration increased sharply with low value of ppd, then kept stable, where it tended to decrease with high radiation. It means that stomata control happens when vapor pressure deficit and radiation reach saturated point to avoid the excessive loss of water. This figure shows the chances of transpiration in different leaf conditions. Stem transpiration went up significantly when leaves started expanding, then kept high value after reaching the full leaf condition. Finally, decreased suddenly when changing to yellowing and falling period. In comparison with previous studies, our result was inside the reported range, and the differences between published values possibly relate to topographic features or climate factors. As summary, we found a new equation which can reduce the uncertainty of transpiration estimates in larch. Besides radiation and vapor pressure deficit are the main controlling factors of safflower density variation. Thank you so much for the attention.